Okay, so today is our 20 lecture. So we are finishing the module on structure. Last time we talked about the 3D structure of chromatin, and today we're to finish with the structure for RNA and protein. I think you are probably already familiar with some of these, like from other biology class. But today we're going to talk about more about the computational and the modeling aspect for these structure. So today's agenda, we're going to talk about the importance, a little bit about the importance of RNA and protein structure, kind of structural assays for probing their 3D conformation, and the computational modeling of their structures. Okay, so we probably know like we can kind of classify kind of structures detail into roughly 4 different types. The primary structure, which are the sequence, like nucleotide or amino acid. The secondary structure is the one that's more interesting. We will talk a little bit more about this one. And we also cover a little bit about how do we predict, how do we model 3D structure, like tertiary structure is one molecules, and quaternary structure is multi-molecules forming complex. So the reason why I mentioned that secondary structure is interesting because it's simple, but it has important function, which we will see in later slides. So for RNA, for DNA and RNA, when we talk about secondary structure, it's the formation of these base pair and the hairpin and the loops, like these diagrams. For proteins, there are 2 key secondary structure, the alpha helices and the beta sheet. So we have seen these in protein structure, we have seen this diagram of RNA base pairing before. These are all driven by hydrogen bonds. So between nucleotide, we know that for A and T, they can base pair with 2 hydrogen bonds, and for C and G, they can also form base pair with 3 hydrogen bonds. For the protein, the alpha helix and beta sheet, these are stabilized also by the hydrogen bond between the C double bond O and the NH from their peptide bond. So the amino acids that are kind of near each other in 3D, the C double bond O and the NH can also induce the hydrogen bonds in the structure. So all these dashed lines stabilize the helix formation and the beta sheet formation. So especially for protein and peptide, people with from physics already know that when you have the peptide bond that connects adjacent amino acids, and this angle, like when you measure the angle between the C double bond O NH and the NH to the next amino acid, to the alpha carbon for the next amino acid, these has to adopt certain degree of angle. It's not like it cannot bend kind of in every direction. There are certain kind of equilibrium or stable angle that it should form. So some of you might have seen the Ramachandran plot before. So this show the phi and the psi angle in the protein peptide bond. And there are only few possible combination of these in nature. Like when you form a peptide structure, if you have the alpha helix, your phi and psi angle should follow some restricted possibility, like around here. The phi angle being about minus 90 degrees and the psi angle also minus 70, 80 degrees. And if you form a beta sheet, like the angle will also be different. So people oftentimes use kind of this combination of angle to kind of validate or to check the quality of a structure. Like if someone gives you a protein structure and the angle between peptide bond and the alpha carbon doesn't fall onto this area, like then you think that maybe there's something wrong with the model. So we do have some knowledge about how protein forms, but you might notice now this will be a theme for this lecture, that we know a lot about protein structure, about how the bonds will form, but we almost know nothing about the RNA and DNA. It's so flexible, and the nucleotides are so homogeneous that we don't really know what's the constraint for the structure. So we will see toward the end that we are already good at predicting protein structure, but we still struggle to predict RNA structure. But sometimes we can get a good model or we can get experimental data that can show us the complex of RNA that are this large or protein that form multi-molecule complex. Okay. So why do we want to understand the function of RNA? Usually we think of RNA as coding for the gene, for the protein. But people found that when RNA form either secondary structure or 3D structure, they have interesting function. You might have seen this from non-coding RNA lectures before, hopefully. So for example, like we think on RNA, RNA can be bound by protein if it has the DNA binding motif. RNA can also be bound by other RNA if if the sequence are complementary. We know that microRNA can regulate, turn off the translation of the target transcript. So imagine what happens if you have a gene where the binding site for the protein and the target for the microRNA can form a hairpin loop. So when this transcript form this secondary structure, it protects the transcript from both the protein and the microRNA. So the protein cannot bind because the binding site is bound to another RNA. And the microRNA also cannot access this because it's forming the hairpin loop with itself already. So the secondary structure can also have a regulatory mechanism. In another example, if some of you maybe study some viruses, we know that the UTR, the untranslated region, both in the 5' and 3', these are not translated into protein. But in some viruses, these regions can form secondary structure or can hybridize with each other. And this can either promote the binding of ribosome. If the 5' UTR can form this hairpin structure can be bound by ribosomes. So that will promote the translation of the viral protein. But in a different situation, like the RNA of the virus might fall onto itself and form this base pair between the 5' and 3' UTR. Then in this case, ribosome doesn't have anywhere to bind. So in this case, the virus can remain dormant and it might replicate its own RNA, but it doesn't produce the protein. So the changing of secondary structure in RNA can maintain the life cycle of the virus. Okay. And we have seen many examples in higher organisms where we have a long non-coding RNA. So this RNA doesn't get translated into protein, but they're still long as thousands or 10,000s of bases. So these long RNA can form secondary and tertiary structure, and their structure can be bound by other proteins. So they can serve as a scaffold for protein binding. Why is it important? Because as an RNA, they can also hybridize with different RNA. They can also hybridize with the genome. So long non-coding RNA with their secondary and 3D structure, they can first recruit protein. Protein can be bound to their structure. And then they can also guide this protein to specific transcript, to specific location on the genome. So RNA has kind of dual function that brings protein to the right kind of location, so that this protein might activate the transcription or might do something to the genome. And when a gene is transcribed, as the RNA is being produced, the part that's already produced, the 5' part, can also form the secondary or 3D structure during the process of transcription. So it can also regulate itself. Once the 5' region is transcribed, it forms a structure. It can recruit maybe more transcription factor or other protein that can also operate on nearby genes. So it also has the ability to self-regulate itself. And you might be familiar with the process of vaccine development. We just got past the COVID-19 period, so we all heard about mRNA vaccine. So when we want to develop messenger RNA vaccine, we we are using either viral gene or some gene from different organisms or maybe synthetic genes, and we are injecting them into our body. So what we want to have for the vaccine is it has to be accepted by our body. It shouldn't be rejected by the immune response. And also we want the ribosome to bind to it, to translate it into protein. So during the process of messenger RNA vaccine development, people actually have to optimize the 5' UTR and the 3' UTR for the cargo. The cargo can be a target gene that you want to express inside human. But you need to decide what would be the 5' and 3' UTR to deliver your gene. So some people take knowledge from human body. We know that some genes like hemoglobin are highly expressed in the blood cell. So we know that the UTR for hemoglobin might have something to do with the high expression for hemoglobin. So some companies kind of attach the UTR from beta globin genes or from some viruses that can infect humans. We know that these UTR are already optimized by evolution so that they can be expressed in human cells. So we sometimes use these sequences in our vaccine, but sometimes we can also try to computationally design a new UTR that forms specific secondary structure that we want, like maybe a secondary structure that can recruit ribosomes. And then we attach that to our gene. Then we have a new, a better vaccine for use. So people want to know how to predict secondary structure for the RNA and the 3D structure for the RNA for this purpose, for example. But in general, what can we do with the knowledge of structure, either RNA or protein? The first step is, of course, to visualize. When we think about mutation, when we think about protein binding, if we don't know the structure, then we cannot imagine it. So the first thing we can do is we can visualize on the screen. Now we see the protein. Now we see the drug. We see where they bind. We can generate some hypotheses. So knowing the structure allows us to also predict binding and interaction between proteins, between drugs, between RNA. Because once we have the structure, we can imagine where they can fit together. We can try to identify a pocket in the protein that may accommodate these drug molecules by looking at the 3D structure. When we have a mutation, we can make hypotheses. If we have a mutation here, in the binding pocket, we think that the mutation might disrupt the binding between the protein and drug. If we have a mutation out here, it might not change the interaction between protein and drug. So knowing the structure allows us to interpret somewhat the effect of mutation or changes on the protein. Okay. And when we have a mutation, if we can simulate the effect of that mutation, for example, we might have one mutation here that changes the amino acid. Then if we can somehow model how the protein structure will change, how the binding interaction will change upon this mutation, then we can visualize the new conformation. We can maybe estimate the new binding dynamics. Will the binding be tighter or be looser than before? And will this affect the downstream function of the protein? So with structure, we can visualize and we can make prediction. Okay. So let's see what kind of tools do we have to understand RNA and protein structure. So the first thing that you probably have heard before would be X-ray crystallography. So we have to crystallize, and that's the most difficult part. We need to crystallize the RNA molecules or the protein molecules. And then we put the X-ray onto the crystal structure, onto the crystal. And you have the diffraction of the X-ray when it enters the crystal lattice. And from these, physics people can calculate if we see this kind of diffraction, if we assume that the molecules are in crystal or in lattice structure, what would be the density for the electron that can diffract or reflect the X-ray into this pattern? So this will be like a physics calculation. Once we get the density for the electron, it doesn't tell us yet kind of what amino acids are where. But because we know that there are 20 amino acids, and we know the side chain for the 20 amino acids, so we can kind of predict or we can model, like we can fit the data. If we see this kind of electron density, which amino acid side chain kind of fit this electron density, because the different side chain will create a different electron density map. So because we know that we only restrict to 20 types of amino acids, we can fit the data with not too much difficulty. So the X-ray crystallography is the gold standard for studying protein and RNA structure. First, because of the good resolution. So you are off by at most plus or minus 2 angstrom on average. And if you go to a database for protein structure, you will notice that more than 80% of the known structure that we have nowadays come from X-ray crystallography. So it's the main technique that people use. But the problem is, like in biology, some protein doesn't crystallize easily. You cannot induce it to form crystal. And even when you form crystals from the protein, like, you probably will realize that that's not a native state for the protein. Like, inside the cell, the protein might be in one form, but when you force it to form crystal, this is not how it looks like in the cell. So even if we get the structure, for many proteins, it doesn't look like the real structure that operates inside the cell. So they feel some discrepancy. But this is the best we can do in terms of studying the 3D structure. Other techniques that worth mentioning, like you might have heard of nuclear magnetic resonance before. So these techniques that try to understand the density of electron, but instead of looking at the X-ray diffraction pattern, we try to induce the movement of electron with a magnetic field. And we learn from mass spectrometry that if you apply magnetic field or electric field like to ions, to protein, that you can study its behavior and you can deduce its property. Okay. And the other technique that's becoming more and more popular is the cryo-electron microscopy. So basically you freeze the protein or the RNA and then you measure its structure. So these two techniques, the downside is the accuracy is less than X-ray. So X-ray is about 2 angstrom. Cryo-EM is about 3.5, so it's 2 times less accurate. But this cryo-EM can work for larger protein. You can freeze protein regardless of the size, and because you don't force it to form crystal structure, it can look more like the native conformation inside the cell. But the heart of all this technique is what you're measuring is the electron density. Like you measure this green mesh of electron density, and then you have to fit like which amino acid falls into this mesh kind of nicely. So similar to when we study mass spectrometry, like we don't really sequence the amino acid. We saw the fragmentation pattern, and we ask like which amino acid combination or sequence kind of fit the observed pattern. So the structure is also the same. We see the electron density, and we kind of guess which amino acid fit into this conformation. Okay. Another source of information that helps us understand larger protein is the experiment like chemical cross-linking and hydrogen deuterium exchange that we learned from the proteomic class. So these techniques doesn't give us the exact structure, but they give us this type of constraint that maybe this W amino acid is nearby this D because of the cross-linking data or because of other experimental data. So even though this experiment doesn't give us the direct structure, this type of constraint, that which residues has to be next to each other, people later found that knowing this information is good enough for us to computationally create the structure for the protein, at least for the protein. So in addition to performing experiment, we can also learn from evolution if we want to understand how proteins form. So if we kind of go back to the early lectures about sequence alignment, when we think about protein from multiple organisms, from multiple species, we can make multiple sequence alignment. We can align the protein from the same family together. And one thing that people notice is that there are certain positions in the protein, like this two position, that in 3 dimensions, they are forming some kind of bond together, maybe an ionic bond, maybe some disulfide bond, maybe other chemical bond. And what happens during evolution is if you have a mutation on one residue, the other residues also have to mutate, because otherwise the protein lose that interaction. So what people found is that during the course of evolution, there are many pairs of amino acid residues where you see what's called a compensatory mutation or co-evolution. So for example, like if arginine changed into a lysine, even though it's still the same similar side chain, but the size may be different, we saw that the other position, the other residue that interact with this arginine also mutate from maybe from aspartic acid to glutamic acid, for example. So we found these kind of co-evolution, the two positions in the protein that change together, that evolve together. And this can be measured by, for example, by calculating the mutual information. So you might not be familiar with this kind of mutual information, but the idea is like if these two residue positions do not interact with each other, mutation in one location shouldn't affect in any way, in any shape or form, the mutation of the other. So if you measure the frequency, how many times is this position arginine and how many times is this position a different amino acid, you should get statistical independence because they don't affect each other. But for position that co-evolve, if you change the amino acid at one location, you're going to see the change in amino acid at the second position. So in this case, like the distribution for the amino acid between these two locations are no longer independent. So we can measure the deviation from independence. And that's one of the ways to do that is called mutual information. Basically, you're comparing the joint probability of two amino acids compared to the product of the probability. Because if they are independent, these two should be equal. But if they co-evolve, these two will not be the same. So people found that by looking at many proteins across many organisms, they can computationally, by calculating this kind of score, identify pairs of residues that co-evolve. And they can use this information to guide the prediction for the protein structure. So now instead of knowing nothing about the protein, now we know that, okay, residue number 1 should be close to residue number 10. Residue number 50 should be close to residue number 100. We have some concepts, some restraints that fix the possible protein structure. And people found that this type of co-evolution not only occur inside a single protein, it can also occur across proteins. For example, if you have two proteins that both of them bind to the RNA in the cells, if you change from one organism to another, like you have some evolution, some mutation on the RNA, the protein also have to adapt, because the protein still needs to bind to that RNA. So when you have mutation on the RNA, the two proteins that bind to that RNA also have some mutation that maintains its compatibility with the RNA. It can still bind to the same location. So people can find patterns of evolution, patterns of mutation that are correlated across different proteins in the same organism. So we not only have constraints for the residue that can help us fold or predict the structure of one protein, we even have co-evolution patterns that help us predict whether two proteins work together or not by looking at these patterns. Okay. So armed with this knowledge, people want to be able to predict the actual 3D structure. So let's see how we are able to do that. So let's start with the assumption. When we see AlphaFold nowadays, we know that once we input the amino acid sequence, we expect AlphaFold to predict the 3D structure. But the first question that you should ask yourself is, how do people expect that is possible? How do people convince themselves that just knowing the amino acid sequence is enough to predict the 3D structure? Wouldn't one sequence be able to produce like 100 different structures, because it should be very flexible? How can we assume that knowing just the sequence is enough to develop prediction or to do anything to get to the 3D structure? So this can trace back to a long time ago, when a scientist named Christian Anfinsen. So he did an experiment. He take a native protein, which has all the secondary and tertiary structure like already formed, and he denatured with urea. So he changed the pH of the condition, and the protein lose its structure. So in his experiment, he asked if he introduced the protein back to the original condition, he removed the urea, will the protein fall back to the original form, or will it fall into many possible forms? And at that time, he found that if the protein is returned back to the cellular condition, you get back the same structure, the same form. So this kind of sparked the idea that if we are focusing on just the cellular condition, knowing the amino acid sequence, it's like knowing just this kind of denatured form. If we put it in the cellular condition, it's good enough to specify the final structure. So there is some determinism that given a sequence, it's going to fall into some defined structure inside the cell. But the caveat here is if you change the cell condition, you change the pH, you're going to get a different structure. So everything that we get from the AI, from our phone nowadays, you have to keep in mind that this is based on the assumption that every data that we are predicting that we use to train the model, we are assuming that protein falls inside a standard or some average cellular condition. It's not true for every condition. But that's good enough for us because we want to understand protein function inside the cell. So if we can predict the structure of the protein based on crystal structure or based on the structure inside the cell, that's good enough for the application. One of the reasons that people want to know what's the explanation for Anfinsen's dogma. How do the cells ensure that given a sequence of the protein, it will fall into the same structure? So some people discover that inside the cell, as the protein are being translated from the RNA, it's not like you produce a whole protein and then it starts forming. The protein has to fall starting from the 5' direction that got translated first. So the fall is a step-by-step process. So from the beginning, you only have a short region of the protein. There's not many degree of freedom. It doesn't fall into many forms. And in many cases, we found that there are other proteins, other enzymes inside the body that help guide the protein toward a particular structure. These are called chaperone proteins. For example, some chaperone might stabilize the 5' position of the protein until the whole protein is formed, until the whole protein is translated. Then the protein can fall into the structure. So this protein can kind of regulate the step-by-step folding for the protein. So the cells kind of have many helps to ensure that each protein molecule is folding into the right way, not in random possible conformations. When people think about what are the forces that drive the protein to fold, in addition to this chaperone that can fix some part of the protein in place, what are the physical laws that govern the protein folding. The people found that the most important force is actually hydrophobic, because proteins are in aqueous, like inside the cell. There are other solvents. And some amino acids, like the one with hydrocarbon side chain, these don't like water, these don't like polar solvents. So hydrophobic force these hydrocarbon side chains to fold together, to escape from the aqueous condition, from the polar condition outside. So this force kind of drives the global flow of the protein. But once the big picture of the protein, like once they fall into places, the forces that lock the protein in place, these are the ones that you learn from physics class, like hydrogen bond, ionic bond, disulfide bond, and van der Waals bond. So these operate on a very short distance. So the overall structure, like once the protein are translated, like it's still a loose protein strand. The driving force is the hydrophobic force. And then at the smaller scale, like hydrogen bond, hydrogen bond, and these bonds lock the amino acid side chain into places. So you get the secondary structure, and you get the more refined protein domain. So in the beginning, like the best technique that we have for modeling a protein structure is that we need to know some proteins that are similar, and we have some knowledge about the structure, and we use that protein as a template, like as a starting point to predict the new structure. So this is called homology modeling. So homology means similar. So basically we start with a new protein sequence. We have to search, to browse against a database of protein to find similar protein. And if we have good luck, like some of those proteins already have a 3D structure, so someone already studied. You can use that structure as a template, and you can fit your new amino acid onto that structure. And then we can optimize the structure. We can move the side chain around, we can move the atoms around, so that the new side chain from our new sequence fits onto the structure. So that's what kind of people have to do in the early days when we don't have any good computational idea. After that, people can be more creative. So instead of kind of trying to find another protein that are similar to our protein, like that's a big condition. Right? If you have a new protein and you hope that someone already studied a different protein in the same family, that's difficult to hope for. Because if you discover a new protein, usually it's a new family in itself. But people found that protein actually have many domains that are conserved as a domain, not as a whole protein. So when you have a new protein and you align to the database, instead of aligning for the whole protein, you can do a local alignment. So for example, the 5' part, like the N-terminus part of your protein, might be similar on one protein family, one protein domain, but the middle might contain a different domain. The C-terminus might contain a different domain. So in this case, using local search, like local sequence alignment, you can identify different templates for different parts of your protein. The N-terminus might be similar to this alpha helix. The C-terminus might be similar to this alpha helix from a different protein. And the middle part might look like two alpha helices interacting together from a different protein. So protein training is the idea that we want to identify partial match to our sequence for the new protein. So we cut our protein into different parts. And for each part, we're going to search for other protein with similar to that part, just to that part. And we're going to take the 3D structure or the secondary structure for that part from separate protein. So it's like building a Frankenstein. You are taking parts from different objects, and you put together to form your new protein. and then you can use this to further optimize the structure. So if you go back about maybe 5-7 years, this is the state of the art for protein structure prediction. You start with a protein sequence and you follow this protein trading idea. So you split your protein sequence into different chunks. Each chunk you try to search for the template for the structure for that chunk. And then you integrate everything together into this kind of Frankenstein of a protein. And then you optimize the structure. And by optimizing the structure, means that we are moving the atoms around based on the law of physics. So adjusting the position of each atom in the structure. So you're going to generate a whole bunch of possibilities. These are all possible structures based on many ways that we can adjust the atoms, the residues on this template. But the assumption is that some of these structures might be close to the actual structure, but some of these structures might be outliers. So after you generate a whole bunch of possibilities, a whole bunch of possible 3D structures, you can do clustering. Basically, you are grouping similar structure together into groups, and there will be structure that doesn't look like anyone else. So in this case, the algorithm can assume that if you have a lot of structure that look similar to each other, this might be a good candidate for optimizing the structure further, because you can arrive to this kind of structure in many possible ways, compared to outlier structure where you have to move the atom in only specific way to arrive at this 3D structure. So this algorithm will repeat this process several times. So it will take a template structure, try to optimize the energy and moving the atoms around, cluster the structure, take the one with many members, use that as the template for the next round of optimization. So the algorithm will keep moving the atoms around. And you can imagine like there are thousands of thousands of atoms inside the protein 3D structure. So there are many possibilities to produce, and this process will have to repeat several rounds until we get to the structure with low energy. We'll talk about the specific term for the energy afterward, but the assumption here is that because the protein should be stable, and having low energy means being stable inside the thermodynamic system. So this optimization process, when it's moved the atoms around, the objective is to reduce the overall energy of the system as much as possible. So this process for predicting the protein structure, if you use this process, what you're going to have to do is you're going to end up with 10 to 20 predictions, because you cannot tell which one is the one. All these final structure, like these 10 or 20 final structure, they're going to have very similar energy, and they're going to be consistent with what we know about the peptide bonds. So all these structures will look equally good in terms of physics. So this is where people found that if we know some distant constraint, like the one that we get from chemical classification or the one that we get from coevolution, if we have information from those, we can compare multiple templates, and we can select the one that fits to the constraint that we have, like to the coevolution pattern that we have. So for example, this is from my own thesis. So crystal structure, like for this family of protein, only show the elongated form, because when this protein form a crystal structure, it adopts this kind of elongated form. But when we perform chemical cross-linking onto this protein, we found chemical crossing or interaction between these residues. And because chemical cross-linking only occurs when the two residues are near each other, in this case, they should be within 12 angstroms from each other. But the crystal structure shows that these residues are 24.5, 36.9, like 36.1 angstrom from each other. So this already tells us that the crystal structure is not correct. Inside the cell, the protein adopts a different structure. So once we perform structure modeling, either with homology modeling or with this kind of de novo modeling, we get a lot of candidates. But because we have this information from chemical clustering, that these two residues has to be within 12 angstrom. These two residues have to be within 12 angstrom. You can select among 20 possibilities which one that are consistent with this condition. So it allows you to select the correct model out of many possibilities. So back then, people want to combine de novo prediction also with some experimental data so that we can filter out the bad models. But those are 6 or 7 years ago. If you fast forward to today, nowadays we have the AI for protein folding prediction. So I'm going to tell you a little bit about how AlphaFold was developed and the motivation. So some of you might have read the news about AlphaFold, and some of you might have seen the reference to this competition, Critical Assessment of Protein Structure Prediction, or CASP, CASP. So this is a community of structural biologists who get together every 2 years, and they create this kind of competition where they will withhold the latest experimental data on protein structure, and they will ask everyone to train their AI based on known protein structure and try to predict the new structure that everyone hides from the world. So basically they keep some structure a secret for a year so that they can use it to test new AI. And even though AlphaFold was famous for predicting protein structure, these communities of structural biologists actually are interested in many diverse problems. They not only want to predict protein 3D structure, they want to also improve homology modeling. Like if you know the template, can you make the algorithm even better? If you know that the protein will form oligomers, like multiple molecules of the protein bind together, can you model that structure? By knowing just the monomer structure, can you model the oligomer formation? And if you have the amino acid sequence, can you tell which residues will be interacting in 3 dimensions? So basically, it's the prediction of coevolution as well. So they actually have many interests in their communities. And what happened between 2018 to 2020 is when Google first entered the competition. So in 2018, AlphaFold version 1, this is the one. This is kind of before the one that you heard in the news. So AlphaFold version 1, when it joined, what it does well was it predicts the coevolution distance, coevolution signal. It can identify which residues in the protein have to be near each other in 3 dimensions. So basically, it hasn't predicted the 3D structure yet, but it predicts the constraint that can be used to find the right structure. And that was 2018. You can see this graph, this is the y-axis, is the accuracy for the predict structure. So AlphaFold was good at the time, but still not that good. About 90 here is the level of X-ray crystallography. So what was the breakthrough that resulted in the Nobel Prize? It was in 2020 when AlphaFold 2 came out. So AlphaFold 2, I don't think we can go into the architecture because there's too many things that I haven't taught you yet. But the idea is that this 2-year gap, AlphaFold people switched their approach from predicting the distance constraint to predicting directly the 3D structure. So let's see how they arrive at this step. So let's take a deep dive into AlphaFold version 1. I think this is still simple enough for us to understand in the context of bioinformatics. So AlphaFold version 1, when you feed it a protein sequence, it's going to do sequence alignment. So Cyblast, like we learned from our second class, Cyblast, it's going to search the protein database for similar protein, and it get the multiple sequence alignment. And from the multiple sequence alignment, this is where it predicts the co-evolution. So it tries to identify two amino acid residues in the multiple sequence alignment that co-evolve with each other. So this is not surprising, right? If I ask you to identify co-evolution in protein, you're going to have to do the same thing. You're going to have to start with a protein that is given. You're going to search to blast for related proteins. You get the multiple sequence alignment, and then you're going to calculate the mutual information across the columns in the multiple sequence alignment. So AlphaFold basically replaces that process with an AI basically that can look at the multiple sequence alignment and then make that prediction itself. And after it made that prediction, it used the co-evolution to determine whether two residues have to be near each other or not. So this distance distribution is a condition on the structure that residue 1 and 5 needs to be within certain angstrom of each other. Residue 30 and 100 needs to be within certain angstrom of each other. And then AlphaFold 1 use those constraints to guide the de novo structure prediction. So basically, AlphaFold version 1 kind of merge bioinformatics from co-evolution to the bioinformatics of de novo structure prediction together into a single pipeline. And that was state of the art in 2018. It's able to predict the best protein structure by just predicting the co-evolution. But the accuracy was still not so good. The accuracy at that time was about 6 angstrom. So imagine, remember that we know that X-ray crystallography, the accuracy is 2 angstrom. The cryo-electron microscope is about 3.5 angstrom. So this AI is still like worse, much worse than experiment. But one thing that AlphaFold version 1 found is that just being able to predict the co-evolution is the single most important factor for predicting the 3D structure. So if you imagine like when you have to optimize the protein structure, you have to optimize many bonds between amino acids. You have to optimize many potential energy inside the molecules. But AlphaFold version 1 kind of show the world that if you have the distance constraint like this, you can forget about many other laws of physics and you can still have a good prediction. So this table kind of shows that if you don't have the distance constraint, your structure is going to be 15 angstrom off. But if you have the distance constraint, you know the co-evolution, whichever other factor that you remove, you're going to get about 6 angstrom of accuracy. So this means that you can throw away a lot of knowledge about physics. But as long as you have the co-evolution to guide the residues pairing, you can get a similarly good structure. So that's the finding, the lesson that people learn from AlphaFold version 1. If you can predict co-evolution, that's good enough to drive you to a good structure. You don't need to worry too much about other things. So in AlphaFold version 2, we are not going to worry too much about the detail of this, but what I want to show is that in AlphaFold version 2, because they already realized that the ability to predict co-evolution is the most important factor. So this entire structure is about squeezing co-evolution. So it takes the multiple sequence alignment, so compared to AlphaFold version 1. Once you perform the multiple sequence alignment, like this is the bioinformatics part in yellow, the AlphaFold version 1 takes the multiple sequence alignment and then just use it. But in AlphaFold version 2, multiple sequence alignment is maintained inside the model the entire way. So it doesn't just use multiple sequence alignment once, it keeps using that information to squeeze details to make the prediction to the protein structure, and it repeats this process about 3 or 4 times. So it tries to learn as much as possible about the coevolution. And this was the one that achieved, I didn't put in the numbers here, but the accuracy is also about 2 angstrom. So it reached the same accuracy as X-ray crystallography by using this architecture. And just to summarize, if you are interested, I encourage you to read the actual AlphaFold 2 paper. But just to summarize for this class, the important factor that AlphaFold 2 found was that you cannot predict the structure in one run. Similar to de novo prediction, you have to predict one structure, you have to do clustering, select a candidate, and then optimize the structure further. You have to repeat this process several times to get to the final structure. In AlphaFold version 2, they also do the same thing. In the first pass, it predicts the preliminary structure, and from that structure, they update it 3 or 4 times. So it's similar to de novo prediction. And AlphaFold 2 people found that that part, that iterative update to the structure, is very important. You have to do that. And you need to apply some constraint to the structure, like the which atoms should be near each other. And this third important factor is that you need a large protein sequence database. And this is needed because when you want to predict the coevolution, if you want to predict evolution, you need to know a whole bunch of proteins. You need to know the evolution basically. So one thing that's kind of hidden from the public eye at the time was the existence of large protein database that occur right before AlphaFold version 2. So it was able to learn about coevolution from a very large protein database. And these 3 factors are good enough to predict the structure that are as accurate as crystal structure. And the interesting thing that AlphaFold 2 people found is that, so in the past, like we used to need some template structure from known protein in order to build our new structure. In AlphaFold version 2, they show that you can throw away the template structure. If the model can learn enough coevolution, the model doesn't need any known 3D structure. It can build a new structure just from scratch. You don't need this template anymore, which is like so very surprising. That coevolution will replace the knowledge of structure entirely. So that's interesting. And nowadays we have AlphaFold version 3. So it's even better than AlphaFold version 2. And the big improvement here is AlphaFold version 2 was focusing on just a single structure. So it's a tertiary structure for the protein. But in fact, when we want to study protein, the protein actually form complex with other molecules. So in AlphaFold version 3, they generalize their techniques so that it can accept more than one protein and it can work with a different molecules. So in this example, from their website, you can model dividing between RNA and 2 different proteins onto the same structure. And they are keeping, they are still improving this to allow different type of molecules and more proteins at once onto the structure. But there are still some limitations. We already mentioned that the data that we use to train this model, the data that we use as a structure of a protein, are from crystal structure. So this doesn't reflect completely or entirely the native conformation. Inside the cell, the protein doesn't form crystal structure. So there's still some discrepancy between what the model predicts and what the protein actually looks like inside the cell. And we know that proteins can change conformation. It can be phosphorylated and change conformation. But from this prediction, you're going to get one prediction per one protein or per one complex. So there's still an assumption that there is only one structure. But inside the cell, there are many possible conformations that the protein can adopt. So that's still one limitation. And this we already mentioned that we can assume that every prediction that we are getting here is based on the average cellular conditions. If you have a different conditions, like you are folding the protein inside your lab, like outside the cells, the prediction might not be accurate because the pH, the other solvent, might be different. So these are still limitations that needs to be improved. But at least for the protein, nowadays we can say that we have a pretty good prediction for the protein structure. I wish we can do the same thing for RNA. So let's see what we can do for RNA. So first, for the secondary structure, we want to be able to predict, given an RNA sequence, which part will form the base pairing, because this can protect the microRNA binding site and can protect protein binding site. So if we are given an RNA and we can predict these base pairing, that's already good enough for many applications. You can identify the state, the regulation of the RNA. But even this is still quite difficult. So there are several ideas that people develop for predicting RNA secondary structure. The first one is also relying on coevolution. So if you have RNA that has to form hairpin in this way in order to have some regulatory function. If you look across organisms and you look at the same RNA, like maybe the coding for the same gene, if these organisms still rely on the same regulatory mechanisms, these RNA positions have to be complementary to each other. So we can still do a coevolutionary style analysis to identify two RNA positions that mutate in complementary patterns. If this is A, this is U. If this is U, this is A, this is C, this is G. If we see this type of complementary pattern, we can predict that these two positions might be base pairing. So that's similar to protein. We look at coevolution in the protein to identify residues that interact in 3D. We can also identify coevolution of nucleotide to identify those that base pair together. The second idea is to try to minimize the energy of the RNA structure. So similar to protein, we assume that the stable form of the RNA or the protein is the one with the lowest energy. But compared to protein, where we know like the forces that control the folding of the protein, we don't really know about the mechanism that guide the flow of the RNA. It's so flexible. Like, what's forcing this C to meet this G? Like, there is no real forces. Like, it's random from our perspective. But still, we can try to search for the base pairing that minimize the energy. So what people first have to estimate is what's the energy for this G and C forming a hydrogen bond in the presence of C here, A here, and CG here. So basically, we try to optimize the energy for each bond given its neighbor. So for example, people have estimated that if you have a bonding between U and G that are in a room area, no base pairing, this is about 5.6 kilocalorie per molar. And if you have a GC pairing followed by C and A that are not paired, this energy is about minus 1.1. It's a base pair, so the energy is negative, it's stable, versus the rooms that's not stable. So people can have to estimate all these energies, and then they try to identify possible base pairing, so that when you sum the energy for the whole structure, this value summed together would be the smallest. But imagine like what type of algorithms you need to do in order to search for all possible pairing of the RNA. There are many ways you can pair the DNA together, RNA together. I'm not going to go into the actual algorithm, but the inspiration is similar to what we have learned before. So the because we know that if we have a short RNA, it's going to be an easier problem than a large RNA. So for example, if you force this A and U to pair, then the problem will be just to optimize the rest of the RNA. So you can do a style of dynamic programming. If you assume that this A and U are paired together, then the problem of optimizing the other will be considered a smaller problem, and you can reuse with dynamic programming. But other people try to make it faster. So with dynamic programming, you still have to consider all possibilities. So they kind of mimic the idea of AlphaFold version 1. So remember AlphaFold version 1, right? We start with a protein sequence, like for AlphaFold version 1. And then AlphaFold version 1 predict which residues should be interacting with each other. So in this model, they mimic the same idea. Given an input RNA sequence, can we train an AI that predicts which nucleotide should be base pairing. So this heat map is a suggestion or prediction from the AI that this nucleotide and this nucleotide should be base paired together. So you have kind of probability that some nucleotide should be base pair, some nucleotide should be kind of alone from the other. But still, like these are just predictions. So you still need to run some dynamic programming to optimize. So based on this probability, which base pairing would be the one that gives the lowest energy and also fit to these predictions. So now we have kind of one more concept to the structure. Some nucleotide should be base pair. Some nucleotide shouldn't be base pair. So your search space, your possibility that you need to optimize is a little bit smaller. So in the end, this algorithm will give you this kind of diagram to show which nucleotide is base pair together. And this is the best we can do for secondary structure of nucleotide. So let's look at 3D structure for the RNA. So similar to the secondary structure, people try to mimic the same success from protein. So similar to protein, like we start with a template-based like homology prediction. So we need to know similar protein, but in this case, you need to know similar nucleotide, similar RNA. And that's even more difficult because if we recall back to the sequence alignment lecture, when we align amino acid sequence, we have a lot of information because amino acid sequence have side chain that have function. So during the course of evolution, similar protein will have similar side chain in a similar position. But if you want to identify two similar DNA or RNA, you don't have much information because each nucleotide doesn't have a function on its own. So if you want to search for template for your RNA, it's already a more difficult problem than searching for a template for your protein. And when people want to mimic AlphaFold, to try to learn from multiple sequence alignment of RNA, and then try to predict the secondary structure, try to predict the distance constant, and try to predict the structure directly. What they found as an obstacle is that compared to protein, which we have more than 200,000 structures, known structure for RNA, we only have 6,000. So RNA structure, the knowledge of RNA structure is severely lacking behind protein. And we know that RNA is already more flexible than protein because there is no charge interaction, there is no hydrophobic force, there is no major force that drives the flow of RNA. So the energy calculation, how the RNA adopt the low energy conformation is not as understood as protein. So this is still a limitation. Okay, so for the last topic, I want to mention another concept, and that's the molecular dynamic simulation. So up until this slide, we think about structure as when we model a structure, we want to model a single structure. But because we know that the proteins And RNA, everything inside the cells can move. Like there are many conformations. So people also want to understand the dynamics, like the flexibility of these molecules. And you have to do that with dynamics kind of simulation. So the concept is simple. Like once we have the predicted structure, like the static structure from either experimental data or from AI, next we want to try to understand how it moves inside the cells or maybe inside the membrane, like in this example. So we want to learn structural flexibility. We want to learn uncertainty of the structure. If it's a binding between protein and ligand, we want to know if the binding is stable. If we allow the protein to move and the ligand to move, will it come apart from each other or will it stay bound to each other? We want to see if protein can change from one conformation to the next with some movement of the structure. We want to see how the protein fold and unfold. Which domain folds first, which model unfolds first. And sometimes we want to monitor which part of the protein is accessible by the solvent. So this might be the part that can interact with the environment. So to run this simulation that you are seeing in this kind of video here, we have to allow the molecules to move. And we have to calculate the energy of the system by monitoring each bonding in the structure. So just to give you an overview of what people monitor inside dynamic simulation. So basically what we are calculating is the potential energy of the whole structure. There is no other energy. The protein is just sitting there. So the potential structure of the system, some of them are caused by the bond between adjacent atoms. These touch on each other. Some are caused by the angle between three atoms. And some are caused by having charge or having polar molecules inside the structure. Some are caused by molecules are near each other and they repel each other. Or there is also some attraction force based on van der Waals principle as well. So what we want to see inside the molecular dynamics simulation is how these force can attack on the molecules and it move them in one direction or the other, and how that small movement can then accumulate into a larger scale reorganization of the protein structure. We want to see how these kind of dynamics that occur naturally inside the cells, can it induce a change in protein conformation? Can it induce a binding or dissociation between protein and ligand? And these are analyzed often on picoscale or nanoscale. So it's very time consuming. Right? Imagine that if you want to measure how protein moves like in 1 second, you need to do 10 to the 9 steps to go from 1 nanosecond movement to a second worth of protein dynamics. So these days, I'm not showing the details, but these days people are trying to accelerate the simulation instead of trying to optimize the atoms sequentially. People are trying to train AI, machine learning model, to try to predict what would you get if you start from this structure and you let it run for maybe 100 nanoseconds. What would be the final structure that you can get? Can we predict this with known simulation data, with machine learning, with algorithms, instead of actually performing the simulation ourselves? So that will hopefully could allow us to not only predict the static structure, but also predict the dynamics of the structure. And that will be useful for biology. Okay, so just to summarize, so these two classes, we talked about the importance of structure, things in 3 dimensions, because everything that we learn in biology exists in 3 dimensions. Even though when we learn about regulation or omics, we tend to think of them as data, as diagrams, but because these molecules exist in 3D, their structure also comes into play when we think about their function. We cover the various functions of chromatids on regulating gene expression inside the cell. We saw today the function of RNA, as a scaffold for protein binding, also as a guide to bring protein to the right location on the genome. And the ability of RNA secondary structure to protect itself from protein binding or from microRNA binding. From protein, you already know a lot about how protein function based on its structure when it binds to other ligands, when it binds to other protein. We learned about molecular assays for solving 3D structure. If you are interested in this, in structural biology, there are many more details that you can study online. And we learned a little bit about the AI that we have nowadays, like AlphaFold, as an example of how we can create a model that can predict protein structure. We learned the principle and what people who develop the AI found along the way that brings them to the current version of AlphaFold that can perform the prediction. And hopefully, we will realize that there are still limitations with all the prediction because they are static prediction and they depend on the context of the cellular state, but it's not explicit. When we get the prediction, they don't have the data that tells us if the structure is based on crystal structure formed in which condition. So the context is still missing from the prediction. So we need to be aware of when we want to interpret the 3D structure for biological purposes. Okay, and that's everything I have for today.